everyone. It's a great pleasure today to introduce Dr. Venkatesh Gopal, a professor of physics at the University of Elmhurst or Elmhurst University here in Chicago, who's currently on sabbatical and has been spending time here at NITMB and has done some really incredible work on Octopus and Odor navigation. So I'm really excited.
Thank you. Well, good morning, and thanks everyone for inviting me. NITMB is a great place to be. I'm always happy to be here. But in particular, there are two people I'd like to thank, Julie and Alasdair. So a year ago at this time, I had no idea about NITMB, and I got an email to attend an open house, and I missed it because I think I was teaching at the time. So I wrote back to Julie saying, hey, can I come visit even though I missed the open house? And she sent me one of the nicest emails saying, yeah, of course, and I'll find you people to talk to. Well, the one person she found was Alasdair. We had a great meeting, and we've been collaborating ever since. So I'm going to tell you, it's a sort of three-part talk today. Part one is the work I've been involved in over the last 3.5 or so years about Octopus odor navigation. But out of that came some interesting questions that Alasdair and I have been working on, so I'll tell you about that. And part three is more, Nature used to have a column called Questions Arising. I don't see it any longer. It's a sort of questions arising. Out of this, there are all sorts of interesting things. There are half-finished projects that I want to finish this year, speculative ideas, all kinds of things. This sort of stuff that you keep in the back of your head. So I want to tell you about that, and perhaps there'll be something of interest for some of you in that. So this slide is not a slide I had planned to show, but this was as I was walking in this morning. That's the Hancock antenna in the background. But it was interesting to see that octopuses were featured even on my walk down Chicago Avenue. And the question, arms or legs? The answer turns out to be arms. So they're called arms. So let me tell you about this problem that I've been working on last 3.5 or so years, odor plume tracking. So before I say a word, let me show you these exquisitely beautiful creatures and how they do this. So what this octopus is doing is trying to find this food source in the dark. So as far as we know, the octopus can't see in the infrared. So there's a slow flow of water going down from right to left on the screen. So watch what happens. So roughly the odor plume is contained in about this area. So watch how it behaves once it gets into the plume. That's odor plume tracking. So it's finding this plume in the dark, not visually assisted. Yes?
Yeah, how big is this?
Oh, I'll get to that, but it's 6 feet by 4 feet, and the octopus is about that big. So several body lengths.
By the way, where does it have olfactory sensors?
It's part of the punchline a few slides in. You want the answer now? No.
No, okay.
Okay. So why should we care? Because many animals, I mean, across the animal kingdom, there's almost no species that doesn't do this. They track odors to their source over great lengths. Sometimes it's to find food, find a mate, avoid a predator. It's a very, very old behavior, doesn't require vision. And some people even think it's one of those grand challenge problems of sensory biology, like how are animals accomplishing this? And sometimes in environments that are very hard to track odors in. So the octopus has distinct hunting strategies, both when it can see the target and when it cannot see the target. Let me show you some of these. This one, there's a small shrimp here, circled in red, and the octopus can see it. And just watch how it sits there, it knows where the target is, gets ready to pounce. And see how it creeps up on the target, encircling it, and then that's that. and it eats the prey by engulfing it like that. So not too many surprises here, but this one, same octopus, same tank, but it can't see where the target is. The strategy is completely different. It knows the target is down here, but it doesn't.
You just turned off the light. Is that the difference?
Yeah,
okay.
yeah. And there are several days in between. They don't eat a lot, so. And it tries to stop all escape. Quite different from the one where it went directly for the target. So this kind of behavior is carried out over hundreds, if not sometimes thousands of body lengths. And we have indirect observations of this, so I'm going to show you some of that. Lots of traps set for fish, most of them which are well out of the so-called floating zone where there's no more light in the ocean. Lots of those traps, there's a lot of octopus bycatch. So they're finding the trap by means other than being able to see them. And here's some data from the fisheries. The y-axis is in tons. So this is the tons of octopus bycatch that are found in fish traps that are often at great depths where there's very little or no light. One of the most spectacular occurrences of this was a submersible doing a live broadcast while just trawling the ocean floor. And it happened to come across at a depth of about 10,000 feet whale carcass. And it was completely covered in hundreds of octopuses. And at this zone, there is no light. So obviously, they've been using, most likely, olfaction to find this target. So why is odor plume tracking a hard problem? Biggest problem is odor plumes are sparse. Right? There are no simple gradients to climb. If you've ever watched smoke, you see that within a short distance, the smoke breaks up into small patches. And so not only is the signal very rare, most of the time you have no signal at all. So in that brief fleeting encounter with the signal, you have to decide how best to move to maximize your chances of encountering the signal again. So especially in these flow regimes that the octopus inhabits, the plumes are unsteady or turbulent. So they break up into patches very, very quickly. So here's a movie showing, this is the plume in our octopus experiment. So this is where the food target is placed. So what we did is seed it with small glass beads and then illuminate the beam with a laser sheet, just so you can see how the plume spreads. So very quickly, I mean, this would be 2 or 3 body lengths, but by the time you get here, there's hardly anything to be seen. Yes?
Is the food stuck on the glass, or is it somewhere in the middle of the
Oh, right. Okay. So the depth of this tank is about 10 centimeters. So it's essentially a 2-dimensional problem. The octopus sort of crawls just above the bottom surface of the tank. So the food is placed on a pin.
Oh, sorry. Okay. So we're looking like top down.
Top down. Oh, I'm sorry. I should have said that. This is a top-down view of the tank. Yeah. So quickly, experimental details. So we wanted a flume that is so large that the octopus has to travel several body lengths. But of course, that's hard to do. I mean, you shouldn't be able to see anything. That's harder to do. And the animal shouldn't have any memory of the tank, so it shouldn't be able to know where the target is, and then second time around just go there purely by memory. And with octopuses, you have the unique problem. They're very, very clever, and they can easily get out of the plume. So a lot of work had to be done to prevent them from escaping.
So what is a flume?
A flume is just a water tank where there's water flowing from one end to the other. Yeah, that's it. So the tank is 6 feet. For zoom, it's 6 feet in that direction, about 4 feet in this direction. Yeah. And this is a time averaged image of the signal. And this is typically, if you look at the scattered intensity and imagine the scattered intensity to be a proxy for the odor concentration, which is not a bad approximation, then you can see how the statistics change very, very quickly as you move away from the source of the plume. It looks like this, and this was really a feat achieved by this grad student, Willem. The whole thing was put in a tent. This work was done out in the San Juan Islands at the Friday Harbor Laboratories. It's part of the University of Washington system. But it rarely snows, and they had a freak snowstorm, and the whole tent arrangement survived. So, testament to the amazing construction done by Willem. And this tank holds about 15,000 or 20,000 gallons of water, which is enormous. And to put that into context, when my son was very young, I went to the fire station almost every day, and I discovered that a pumper truck has 50,000 gallons of water in it. So this is roughly half a fire truck. So our subjects are these red octopus, and they're found fairly abundantly in the Friday Harbor lab region. And the way they would get them is very interesting. Octopus is very curious. So the divers are the grad students, and I think this is Willem. You just place a bottle with interesting things in it, like Lego bricks or little matchbox cars. And then you leave it there, and 2 or 3 days later, octopuses like to have little secure dens like this, but they also want it to be filled with objects that are interesting to touch, curious, not what they find outside. And they go back, and sure enough, there's an octopus in every one of these bottles that they've placed. So we did 2 sets of experiments. First is a single target experiment. And what did we hope to see? Most often, across species, you find a single strategy being used, and it's called odor-gated rheotaxis, which is a fancy name for when you encounter the odor, go upwind or upstream. When you lose the odor, go crosswind to pick it up again, and then go upstream as soon as you encounter the odor. But of course, different animals have variations on this. Some of them, like this one, they go to one edge of the plume, overshoot, then turn in the opposite direction, overshoot, and in that way they find their way to the source. Some of them, like crabs, will track the edge of the plume. So there are a variety of strategies. But overall, it seems like odor-gated rheotaxis is very popular. So here's a single target experiment. That's Willem coming in to put the food in. Octopus, they are very shy creatures. They barely leave the den. I'll say a bit more about that in a minute. Sit still, and the flow velocity, I forget, it's maybe like 5 centimeters per second, but if you calculate the time delay between when you put the food in and when it starts moving, that matches up nicely with the flow velocity, which is another hint that, yeah, they're sensing odor. They didn't pay attention to the fact that he disturbed the water surface, because they are also very sensitive to vibrations. And now it's made contact with the plume. You saw that lunging behavior. So these are the highlight reels. Octopuses don't like to feed. So you transfer them from their holding tank, and they don't like a change of environment. They would hide out in their den for 1 to 2 days. Then they would come out from the den and then just circle the tank for another day or a day and a half. And then finally, they wander about the tank a little bit. They don't like to leave the security of the walls. And then they might feed. So we have 100 days of octopus videos. 100 days of videos, 2,400 hours. And out of that, it fed only 32 times. So it's a rare event that you're looking at. Well, now we did a 3-target experiment. The food is placed in one of these 3 positions randomly. And we observe the same behavior. It gets into the plume and immediately seems to know where to go. So they search around a little bit. And the reason we think there's no memory involved is they never went to previously known locations just to see if there was food. Each time it was a trajectory like this.
Sorry, question.
Yes.
Is there anything in the control positions ever that would not be food, but like a Lego block or something?
No, but so the piece of here.
With the
Yeah.
curiosity you mentioned, like how do you distinguish this from true?
Yes, so all the food is put up on a little peg like that. So the pegs are there at all positions. The only thing that changes is there's a small piece of shrimp. It's like 5 millimeters. And so we see these characteristic motions where the octopus starts out, it wanders about, it hits the plume, and suddenly its behavior changes. And very often you can see one of the arms lunging towards the food. So at this point, it's good to talk about why octopus. So many animals have been studied. What's so special about the octopus? The octopus is really interesting because there's very little top-down neural control. All other animals, a lot of what happens is top-down. The brain's even simple thing like picking up an object. The brain sends a signal, you make contact, there's feedback. The brain decides what to do with that feedback, but it's all run from up here. But in the octopus, the arms are more or less autonomous. There is a central brain. Some signal is indeed sent there. But very often, the arms are just behaving. They're sort of brains in their own right. So there is evidence where the arm gets cut off. The arm will continue to forage for food on its own. But then, when it gets the food, it then tries to feed a mouth that's not there. So it's not aware that there is no mouth to be fed there. So in that sense, the entire control algorithm seems to be completely different. And that's what makes it very interesting. And that's what makes it surprising that the octopus would also use odor-gated reataxis. So what we've, yes?
Tank is a rectangular tank.
Yes.
So how are you maintaining that the flow is just constantly going in this direction?
That was hard to do. But there are there's a set of, I'll show you the picture in a second.
But it was.
It works. Okay, you're happy
I mean,
with
that was the main thing that I, just.
Yeah. So there's a plate with a number of holes, and then you had to tune you had to adjust the pump rate so that you would maintain a constant flow rate and a constant height.
Because if it was a really changing flow, it would probably be very, the octopus would have to be responding to that.
Yes, so the flow was steady throughout the... So it always chose the beta target, very significantly, it chose the baited target over the unbaited target, and very significantly, it almost always went upstream to direct the target. Only on 1 or 2 trials, it accidentally hit the target while going downstream. So now, the other interesting thing about octopus is that very little is known about them. So for a long time, there are 2 little structures. So I have a tiny octopus model here, not very realistic, but there are 2 little structures here called the olfactory pits. And for something like 80 or 90 years, people thought, they named them the olfactory pits because they thought, okay, that's where olfaction happens. But what we saw was these suckers that cover the arms, there's about 3 orders of magnitude difference in the surface area covered by the suckers and that covered by these 2 tiny little pits. And so what we found, the main finding we had, the suckers are the primary chemosensory units. Because we could see in the videos, as soon as any of the suckers entered the region of the plume, immediately the behavior changed. And that arm would sometimes pull towards the direction where it encountered the chemical. So within the plume, it's absolutely beautiful, the sort of trajectories you see. But almost all of them are clearly odor-gated reataxis. Because you see, this is roughly, this is roughly the mean area occupied by the plume. And you see the majority of trajectories are in there. There were a few lucky strikes from elsewhere. But otherwise, it encounters the plume, turns upstream, and finds the food. These are some of the trajectories. These are, in fact, all the trajectories. That's the plot of the heat map of the plume. This is light scattered off glass beads, so it's very crude compared to the odorant. The odorant is probably, there's much more gradation, but we are unable to see that. But at least it's a rough estimate of where the plume is. And you can see all the interesting zigzag trajectories. There are some where it comes in, hugs the edge. You see a whole variety of trajectories. Here are even more. This one, for example, is a lucky strike. It comes in, happens to be right close to the source, encounters the plume, immediately turns and gets the food. Yes?
Shape always identical?
No, because the flow is unsteady. This is just a rough approximation to the strongest areas of the plume. Yeah. Because the flow is changing all the time. One of the problems in this experiment is you can't map the flow simultaneously with the animal behaving in it. This is an interesting question. So David Scheel is the octopus expert on this project, and he asked, is the octopus a bilateral or a radial animal? So we have bilateral symmetry. There's a midplane. The body is roughly symmetric about this. So is the octopus. So if I were to stand this way and you were to call my name, I would reorient to center you on my midplane, and then I would answer your question or whatever. The octopus behaves very differently. Although it has this body symmetry, if the stimulus is to come from here, the octopus doesn't reorient itself. It just goes whichever way the stimulus it is encountered. So that was very interesting. We found that the octopus behaved more or less like a radial animal. For example, this is a mouse here, and the mouse is tracking an odor plume, but it always tries to orient itself symmetrically it tries to equalize the signal on both sides. So we found this is a heat map for directions that the octopus would travel when there's no food. There's only a slight bias in the arm directions, but otherwise the octopus is behaving largely radially. So we saw clear signals of odor-graded rheotaxis and plume tracking behavior in the octopus, and that was the main takeaway. But the interesting thing was this is the first laboratory observation of odor following in octopus. So in the immortal words of Monty Python, now for part 2, it is something completely different. Questions arising from the octopus. So this is the problem Alasdair and I have been working on. So when I looked at this, my interest was, so I'm interested, as a physicist, I want to look at the more mechanical questions. So I was curious as to why all these suckers would be circular. Is there some advantage to having a circular shape? So does it confer any advantage to have ring-shaped sensors like this? So I had once heard a terrific talk by Gilles Laurent where he demonstrated how nature will often find the same solutions in very different niches. And so I looked around a little bit to see if there's any other evidence of circular suckers, and I happened to come across this paper which showed that the sand scorpion behaves like a circular sensor. So what the sand scorpion does is quite interesting. It hunts at night, sits on the sand. Its prey are small creatures like, say, crickets, and we heard also that 40% of its diet consists of other scorpions. So if there's a slight motion, then it sets off a surface wave. Now, it takes about 20 centimeters or so for that surface wave to become roughly a plane wave. So when this plane wave travels across the legs of the octopus, the legs have what are called slit sensillae. So you can imagine a sort of accordion paper folded like this, say, that can be squeezed like an accordion. And these slit sensillae are enormously sensitive. They can detect sub-nanometer vibrations. So when the wave passes across the slit sensillae compress, they compress and expand, the neurons fire off. So each one of these slit sensillae are connected to what are called command neurons and then an inhibitory neuron. But there's also other complicated wiring. And this paper was very, very hard to read. But finally, I realized that this is some sort of vector addition that they're doing. So one of my undergrad students needed a senior project. I said, why don't you simulate the system, but just make these simple, what we call sensory dipoles. So let me explain how this idea works. So let's say you have two of these dipoles. That's one dipole, that's another dipole, right? So let's call the orange one A and the red one B. See, here comes the plane wave. It hits this dipole, so this dipole starts counting, it starts firing off pulses. When that plane wave reaches its counterpart on the other side, that sends the inhibit signal and tells us to stop counting. So let's say you've been firing pulses at a constant rate. You count those pulses and you call them T1, say. Now, this other dipole has the same pattern. So whichever command neuron is hit first, that's the one that starts counting, and the other one becomes the off switch. But now this wave has a shorter distance to travel before it hits this guy. It hits this, sends the off signal. So you've got 2 times stored in each of these dipoles. Now, if you just make 2 vectors, one vector, so the rule is make a vector, starts at the center, points in the direction of the cell that was hit first, and the length is proportional to the time between the on signal and the off signal. This is just good old vector addition that we teach high school and college students. You add up these vectors, and it points reliably in the direction of the source. So that's when Alasdair got involved, and he's derived some really beautiful results that even if you put in some jitter here in the leg position, this still works almost perfectly to point towards the source. So this is also well known in biology. It's called population vector encoding, but this is a much simpler model. It doesn't involve neurons. It's just on-off. So this is some of the simulations my student has been doing. So what we did is put in some jitter in the leg position, and it's still a very, very good indicator of where the disturbance is coming from. So now so Alasdair and Axel and I hope that we're almost at the finish line for that problem. But it all started with, the problem we're looking at is the Octopus, which is looking at turbulent odor plumes. Can we extend this idea to turbulent air or water currents? But, there are no nice plane waves in a turbulent flow. So the sand scorpion is dealing with plane waves on the sand. But where are the plane waves in turbulence? Turns out, on small time scales, even if you had, say, a corrugated odor plume front moving this way, on very short time scales, say millisecond time scales, the dominant motion is advection. There's hardly any mixing or stretching or anything going on. Everything is just going in a straight line, just like plane waves. So our hope is to exploit this to see if these circular sensors could work for turbulence. So here's an experiment I did, but I've since dismantled this. The idea was to do a hybrid experiment, get the signal from hardware or nature. So we have smoke plume in a laser light sheet, and now implement the circular sensor in software. So the scattered light would be a proxy for the odor concentration. You simulate an array of circular sensors, and then see if the same population coding algorithm could work to help the circular sensor find the source. So this is an experiment I hope to do.
Sorry.
Yes.
To understand, so the circle you're talking about is the sucker. So where are the sensors exactly in sucker?
As far as we know, the rim of the sensor sucker is densely covered in olfactory sensors.
So so around each sucker?
Yes. Yes. Yeah. Thousands. Yes.
But I would somehow need the opposite ends of the circle to be talking to each other. Is that right? Or...
We have no idea, because people have only just started looking at the neural architecture of these suckers. And the first glimpse we've had is there's just dense wiring. We have no idea. It looks like everything's connected to everything else.
So if I take the dipole array, but I don't have things connected across diameters, but I have them connected across, random connections or something like that, but they sort of know who they're talking to, could you still work out what direction?
I don't know. Right now, the dipole is the simplest tractable system to work with. And then we can make the wiring more complicated. So based on that, I'm interested in general in the problem of how much information you can extract out of turbulent flow fields. So one thing I had done some time back was build a really simple whiskered robot. So as a physicist, the problem I'm interested in is A, how physics places limits on animal sensory systems. And second, to build hardware models to understand what computations the brain might be doing to extract information in these situations. So in this whiskered robot, really simple, you have little strips of plastic that represent the whiskers. They flex in the wind. They have tiny magnets at the end and little magnetic field sensors. So as the whiskers blow around in the wind, the magnetic field changes, and that's a proxy for the displacement of the whisker. So this was a long time ago. This is the first version that my students built. They built it all in a week with Lego. So what we have here is we have a fan here. There's a fan blowing. Here's the giant robot, and there's 4 whiskers, and it tries to find the fan. And the algorithm is very simple. It compares the standard deviation of fluctuations on both sides and turns in the direction of the greater standard deviation. And you tune the amount of turning with the potentiometer just to adjust the potentiometer. Try a few times and set it to the best position. You see that it finds the fan. But then it occurred to me that the same algorithm would work even for avoiding an obstacle. So if you have an obstacle in the flow, then there's a wind shadow. And the same algorithm, turn in the direction of the greater standard deviation, helps get you out of the wind shadow and perhaps back into the flow again. And indeed, even with this giant, clumsy robot, that is what happens, which was a nice surprise when it actually happened. This motion reminds me of a title of a book that is Simple Heuristics That Make Us Smart. And it really appears to be smart behavior on the outside, but it's not. It's just this really bare bones, simple algorithm just turning in the direction of the greater standard deviation. This is version 2. We've done the same experiment with just better starting. So there's a fan here. We start our robot off from different positions, and it reliably finds the fan from every position. And this always reminds me of a fly. If you've ever watched a fly walking on a table trying finding food, it's very reminiscent of that sort of behavior. But each time it's finding the fan. So the stop-start motion is because when you move, the whiskers move due to inertia. So you have to wait for the inertial motion to die down, then sense, then decide where to go next. But then I wondered, what about the mean? What happens if you compute the mean instead of compare means on either side instead of comparing the standard deviation? Turns out the mean doesn't work as well. It works better than this. In this experiment, the robot just spun in place everywhere. But another version of this experiment that we did, the mean did work, but it wasn't as successful as the standard deviation. So my hope is to build a faster robot. We've built a little bit of version 3. And I'd also like to subtract inertial effects from wind effects so that we can have a robot that's on the go all the time. It's not stopping for the whisker motion to die down. But these are the sort of problems. It's a little bit like number theory problems. You can state them very simply, but to actually do them takes a lot of time. But a real surprise that came out of this was something I like to call flow signatures. Because I wondered at this time, when wind flows across some kind of terrain, surely the terrain imparts some sort of signature. The flow statistics must change depending on the terrain that the wind is blowing over. So I had a terrific student who set up these whiskers, and Lego is a common theme, in 4 different environments. And we had a fan blowing some air just because when the wind wasn't blowing, we didn't want dead time on the sensor. And very interestingly, just looking, so he built a big matrix of, I think, 15 different parameters for the wind and made a neural network. And the neural network converges very nicely. It recognizes each. It seems to be acting like a classifier for each of these different environments just from the knowledge of the motion of the whiskers. But one of my friends, Mitra Hartman at Northwestern, had said, Everybody hates neural networks. But the idea is you've just exchanged one black box for another. So I tried to see if I could get at it through some statistics. And I calculated what's called the Hurst exponent. It's something similar to a fractal dimension, measures the roughness of some signal. But the signal, again, converges very nicely. The roughness of the signal is different for different environments. So could animals be using this as a sort of look-ahead strategy in the dark, perhaps? I don't know. But this is very interesting. This is definitely something I'd like to pursue further. Another question I was interested is, across the animal kingdom, you see animals with bilateral hair-like structures, right? There's a body midplane, and they have some sort of whisker-like structure symmetrically placed about this. And of course, whiskers have antennae because many of them do active sensing of chemicals. But I was wondering if there was a purely mechanical reason for them as well. So in the problem of odor following, suppose you have an odor source, which is sending out odor packets in a turbulent flow environment. There is a surefire way to find the source, 100% successful. But of course, this is a lattice model and so on. And it's a simplified version of what happens. But the strategy is pretty simple to state. Just move to the position from which the last packet came from. Now, if you do this, you can see this is a neat little paper. It's odor-gated rheotaxis. You go upwind, you lose the odor, you cast crosswind, and you find the odor, you do the same thing, and you find the source. However, this model has only one free parameter. That's interesting. Now, odor following, you want to find the source, but you want to expend as little energy as possible. The thing that costs energy is the cone in which you search, right? So you can, of course, be successful if you had a huge cone in which to search, but it's very wasteful. But it turns out the only free parameter is the eddy diffusivity. That is, if you had two bubbles that started off with the source, they diffuse out in the sense they would be on a sphere of some radius as time goes by, or as the square root of time goes by. Now, what if whiskers could measure the turbulent microstructure and decide how wide that cone is? That would be cool. But that's just an idea I have, but it's something I'd like to see if this is true or not. There are other flow sensing hairs. This is a strategy across nature. Another system I was interested in is the cricket circle system. So crickets have a visual blind spot over here. They can't see if a predator attacks in this angle. So what they've evolved instead are these two things called cerci, or cerci, and they have very fine hairs. So if a predator approaches from behind, these hairs vibrate and they cause the cricket to initiate an escape response. So I built a hydraulic sled to simulate a predator, and I almost set up this very nice experiment to see whether you could detect the angle at which the sled was coming from. So I had artificial whiskers, you could change their angle. But very sadly, I got a new student, so I had a terrific undergrad who built all this and graduated. So the next student asked him to clean up the table. He thought that meant remove everything. So when I stepped in an hour later, half the experiment was gone, and I haven't since gone and built it back. But if someone wants to volunteer to do that, I'd be happy. So the story is there are so many ways to, there is so much information being carried on turbulent air and water currents. And there are so many ways to extract this information. But one of the main techniques you see in nature to extract information from a turbulent plume seems to be just a little hair sticking out into the wind. And the vibrations of that hair, or perhaps an array of them, and the correlations between them, seem to carry lots of information. So it's a terrific place to explore. So thank you to many, many people, but really, Willem is the fantastic grad student who did this amazing experiment. I don't know how many nights he spent without sleeping, waiting for the octopus to feed. He more or less single-handedly did this absolutely amazing experiment. So thank you, Willem. But there are also many other people at University of Washington, Elmhurst. Thanks, everyone. Yes.
I was really charmed by your standard deviation idea of sort of going to the source that way. And do you have any speculations about a neural implementation of something that measures standard deviation?
No, that's
wondered
why I'm here. I hope that someone on the more biological side could point out.
Yeah, because you need to have some memory. I mean, you need the
Yes.
mean, right?
Because I've been thinking of electronic implementations, and you have to have some kind of memory unit that's saving the squares of the fluctuations.
Yeah.
Which is not hard to do, but I don't know what does that in the brain.
Okay, me neither.
Yes.
A question about the this turning up wind. So it looks like they don't turn up wind on the first encounter. Like, is there a sort of majority voting where it decides to start turning up when enough of the legs are inside the plume?
Majority voting is the phrase that popped into our heads on watching these videos, because there are some videos where you see two arms, each one pulling in another direction, almost as if they're playing tug of war with the center of mass of the octopus. And then one of them seems to win. But we don't know what the algorithm is. I mean, I feel like this has just opened up more questions than it has answered. Yes.
Do you know if they can do this in 3D also? So if you had like a big tank and a food source in the middle.
Yes, definitely. But this is purely human limitation. 2D was hard enough. 3D is harder still.
So are octopuses good swimmers? I guess
yes.
They can. Okay.
But good point. They tend to crawl much more than they swim. They don't like to be unbounded on all sides. They like to hug a surface. And so very often what they do is, even in high turbulence flows, high speed flows, they will crawl along the surface more than they swim.
Mm-hmm. And I mean, are sort of fish doing a similar process? Like if I had a
yes, there is a paper about dog sharks following odors. They also do odor-gated rheotaxis.
Mm-hmm, mm-hmm. But you think it might be a different mechanism because they're more sort of top-down?
They're more top-down, and the sensors are not so distributed. Here, you have a distributed sensor array that's continuously reconfiguring itself without top-down control, without as much top-down. It's a completely different way of playing the game.
Can you sort of like cover up the olfactory pits or something? Can they
yes,
find
that was a slide I deleted. Because one of the obvious questions is, what happens if you ablate the olfactory pits? We couldn't do that. Because to go in and do that kind of work takes even more time.
Mm-hmm.
But we're somewhat confident that wouldn't have told us much, because we see that the octopus reacts as soon as the tips of the arms enter the plume. The signal hasn't even reached the olfactory pits.
Mm-hmm. Mm-hmm.
From all the videos, it looked like arm behavior was driving the motion of the center of mass.
How do you know if the tip is in the plume? You can't visualize the plume.
No, we can't. But we have a rough idea where the plume is.
Uh-huh.
So we roughly know that the arms are.
And is that a technical thing that you can't visualize
yes, it's a deep lab cut problem. So we tracked the arm motion is sometimes the tips of the arm sometimes move too fast. So it's two things. There's not enough light, even though you have very bright infrared lamps. So when the arms move very quickly, there aren't enough bright pixels for deep lab cut to reliably track the arm tips.
What is deep?
Oh, I'm sorry. The automated tracking program.
I see.
Yeah.
Wait, sorry, no, I guess ideally you'd want to visualize the plume and the octopus at the
same
time, right? But like you cannot.
That's a really hard problem. That is a hard problem, because then you'd have to have a very bright laser. Then there's clearly a visual signal. Then there's also the question of, could you damage the octopus's eyes with a bright laser beam? So yeah, that would be the ideal experiment to do, and we have thought about that a lot.
Mm-hmm, mm-hmm.
But the other thing is the laser intensity goes up a lot if you want to image a dye.
Mm-hmm.
You'd have to put a lot. This was like a 40 milliwatt beam. that would be a much more powerful laser.
Could you somehow like Schlieren image the tank and see the...
No, that's a beautiful question. And in particular, I like it because I've built at least 2 Schlieren imaging systems. Schlieren will measure small changes in the refractive index. In seawater, in which these experiments have to do, there are lots of changes in the refractive index. Now, if you wanted to put in, so Schlieren will do the smallest it'll do is a 0.1% change in the refractive index. But in olfactory terms, that's a lot of odorant. To get that change, you'd have to put a lot more. So that would be a signal that's not biologically plausible.
But it wouldn't just allow you to sort of track the flow field, and then you could assume
I
that
don't
sort of
know.
everything.
So the trouble with Schlieren is, so there are 2 kinds of Schlieren. One is, what do I call it?
Maybe not everybody knows what Schlieren is.
Sure, why not? Schlieren was discovered by Robert Hooke, the guy of Hooke's law. So if you have a source here sending out light, and then I have, say, a lens here, then all the beams get focused to a point, say, yeah? And now, if I have a circular source, at this point, I'll have a circular image. Now, there's a very clever trick. You can put, say, a razor blade or something here and block off half the circle, and then let it go on to hit a screen where it forms a big circle. Now, if I have a change in the refractive index, say there's a candle which is sending hot air, right? Then these beams are going to bend differently. So maybe the beams now that would have gone this way are actually going up. So they end up on this part of the image. So places where the refractive index has changed, the intensity changes. And the intensity change is proportional to the gradient in the refractive index. So if it becomes denser, and you have a positive gradient, it becomes brighter, or the other way around, I forget. Now, this, of course, you can't do with the Octopus. You need a gigantic mirror or lens to do this. But there's a clever new way of doing Schlieren that came about in the last 20 or so years. You have a target, and you image the target. So if I can have a big screen, the size of this screen, with little dots on it, and I image that. And you've all seen this. Suppose you're inside a warm room, the door opens, it's cold outside. You see the heat waves going out. So if there's a change in the refractive index, the positions of those dots would change, and you can have a program track those dots. So yes, in principle, that could be done, but it's hard. I built a Schlieren system to look at odor plumes for Drosophila larvae. And that took a while to do. So scale that up but yeah, in principle, yes. Yes.
Do you add just like a dye to the water and see how it spreads? I'm thinking of Chicago River and St. Patrick's Day.
Yeah. You could. Yeah. We couldn't find a dye that wouldn't mask the. We didn't try too hard because we got this experiment going.
Yeah.
But yes, that could be done.
Something odorless.
Yeah. That also fluoresces. But then the trouble is we'd have to get something that fluoresces in the IR so you don't have to shine a laser. And okay, now I remember why we couldn't do that. The IR sources had to be very, very, very bright to get any fluorescence from the dye. That was the problem. We did think of this.
But I guess you could do it in higher light levels and then just assume that it's sort of similar.
I guess, but I remember, this is a few years ago, but I remember there were too many lights involved if we wanted to get any signal out of it.
You had a slide about some neural nets there with a matrix.
Oh, yeah.
Matrix. Can you
yes, sure, yeah.
Go there once?
Yes.
Yeah. What is the data being fed here to the neural net? Just
So,
at a high level.
yeah, like a mean, a standard deviation, the difference between the first and fifth value. So this student, he came up with a matrix of 15 different things that he just created out of that time series. So just take a time series and think of 15 things you could do with it. So you window the data. Let's say that's the whole time series. Break it up into little epochs, and then find things. So in this epoch, say you calculate the mean, you calculate the standard deviation. So you build a vector that has those numbers for this epoch. And then you build feature vectors for each epoch. And then you just throw those feature vectors into the neural net. That's what he did.
And let's thank Venkatesh.